So let me start. For over 100 years, light detection in the eye has been synonymous with its absorption by retinal rods and cones for the purpose of image vision. For the past 20 years, however, the largely subconscious non-image vision has also come into prominence, such as photoentrainment of the circadian rhythm and pupil light reflex, together with its associated photoreceptors. What's more, yet additional photoreceptors now appear to be in the eye. And in this talk, I shall give a quick overview of the established ocular photoreceptors and the signalings. Because of time limitations, I shall focus mostly on concepts rather than detailed experiments. So first, rods and cones. This is a rod cell. In darkness, a steady current enters the outer segment through open non-selective Hcn channels. This so-called dark current depolarizes the cell and maintains a steady release of glutamate at the synapse. Light closes these channels, thus stopping the dark current, causing a membrane hyperpolarization as the light responds, which then reduces or completely stops the glutamate release. So how does light close these channels? Well, in the early 1980s, our group, together with Yevgeny Fesenko's group, managed to sort out 15 years of debate and controversy into a coherent picture. So let me summarize. This is the rod outer segment membrane, and these are the light-sensitive channels found by the two labs to be open in the dark by cyclic GMP acting as a ligand. Light photoexcites rhodopsin, which via the G protein transduction stimulates the phosphodiesterase to increase the hydrolysis of cyclic GMP. As a result, the level of cyclic GMP falls and the channels close to produce the light response. These active components are then deactivated or inactivated via elaborate mechanism shown in blue here. Now, calcium is also important. In the early 1980s, we discovered this steady circulation of calcium at the outer segment, in through these light-sensitive channels and out through this ubiquitous sodium-calcium exchange in cells, which pumps out internal calcium. Later, Peter McNaughton's group in Cambridge, England, showed that potassium is also involved in this sodium-calcium exchange. At least it is the case in retinal photoreceptors. We discover a membrane current associated with this exchange activity. We call this exchange current. It is an inward current because the stoichiometry, as discovered by us, was one net sodium entering or one net positive charge entering for each calcium leaving. Now, the discovery of this exchange current is wonderful because it allows us to monitor the calcium efflux with electrical recording, which is very convenient and is in real time. And so, as such, what we found was in the dark, the influx of calcium stops because the channels close, but the efflux continues unabated. We found that the efflux continues as if there's no light or dark. It just keeps going. And it depends on only the calcium concentration. As a result, the internal calcium concentration is drained. The free calcium goes down in the light. Next, what we did was to use a simple trick to simultaneously stop the calcium influx and the calcium efflux in the dark, so that it's going to be clamped and it's not going to change in the light. And what we found under these conditions, the light adaptation, normally shown by rods to steady light, as indicated by this progressive decline of the response from a peak level to a steady plateau level, disappears completely under this condition. So the light-induced decrease in internal calcium is a primary trigger for light adaptation. Over time, biochemists figure out what calcium is doing inside. Namely, the calcium mediates multiple negative feedback mechanisms. One is on the guanylate cyclase, another on the termination of the active rhodopsin activity, and a third on the gating of the channels by cyclic GMP. In the mid-1990s, we decided to quantify each of these feedbacks so we can weigh their relative importance in light adaptation. Just a few words first about the methodology. So we use a standard suction pipette recording method developed by Baylor, Lamb, and myself in the 1970s. This configuration consists of the outer segment inside the recording pipette. Another configuration consists of having the outer segment outside the pipette, so we can subject it to different bath solutions. And this method turned out to be extremely useful later for my lab to figure out phototransduction. Finally, here's another configuration with the outer segment inside, but then we truncate the cell to give rise to an open-ended rod outer segment under recording. We can dialyze different solutions from the bath to the inside. We use this what we call single-cell biochemistry method to really measure different biochemical parameters for phototransduction. And then we put all the measurements together to do mathematical modeling. And as such, we concluded in the mid-1990s that this feedback on the calcium, by calcium on the cyclase, is very important in the dark for stabilizing the dark current and therefore reducing the signal background or the signal baseline. Also, with background illumination at low and intermediate light intensities, this feedback is critical for light adaptation. With progressive increase in illumination, this feedback on the termination of rhodopsin begins to kick in, and it becomes progressively important with increasing light. This feedback on the gating of the channel by cyclic GMP is basically unimportant under all lighting conditions. Now, let me just say that, let's see. Oops, sorry. So, work from our lab and also that by Benjamin Kaup's group, Steven Siegelbaum's group, and Bill Sagata's group, we now know a lot about these channel subunits, the molecular subunits making up the channel. We call this, by the way, CNG channels, cyclically irradiated channels. And we also know about the modulation of these channels. As such, the picture of rod phototransduction is quite complete. Now, let me say, what about cones? You know, early on, some people thought that phototransduction in cones uses a different mechanism, but in due course, our lab showed that in fact, almost exactly the same picture applies to cones, except for different rod cone isoforms found by others that correspond to many of these proteins here. And these differences together to some degree with the morphology, makes the cones much less sensitive to light, typically about 100 times, and also gives rise to faster response kinetics. Now, there's another difference between rods and cones, namely that unlike the rod pigment, which after formation does not go back to naked opsin and chromophore, the cone pigment actually in complete darkness can spontaneously dissociate back to the naked opsin and the chromophore. This was first described by Yoshizawa and co-workers in Japan, but later on, we also have done a detailed study of this phenomenon. And this work 15 years ago was done by Vladimir Kefelov while he was a postdoc in my lab, who's now, of course, a professor at UCI. Lucky you people, by the way, because Vladimir is very, very good. Okay, so most recently, our lab, in conjunction with Carter Cornwell's lab, we work on goldfish, and we found that the red cone pigment in situ actually has a substantial dissociation. 30% of the pigment dissociates back in the naked opsin and chromophore, even though for the green and the blue pigment is very minimum. Now, because naked opsin, whether from rhodopsin or cone pigments, naked opsin has conserved activity to activate phototransduction or the phototransduction process, and as such would lead to, especially in the case of red pigment with so much opsin, in aggregate is going to produce a fair amount of light adaptation, not light, but equivalent to light adaptation, making the cells less sensitive to light. And we think that this may be the reason why red pigment, in compared to green and blue pigments, are quite a bit less sensitive. Maybe not animal, but definitely in some of them. Okay. Now, so the bottom line is that for both rods and cones, this mechanism essentially describes the phototransduction process. It provides a quantitative and detailed foundation for understanding not just normal transduction, but also under disease conditions, namely practically every protein shown on this scheme and then some more, when mutated, would cause blinding diseases. At the same time, this picture here provides the most, I wouldn't say the most, but it's near the most comprehensive understanding of Gpc signaling over the entire body. Now, let's see. Let me now introduce another representation of rod phototransduction as shown here. Well, 35 years ago, or almost like 40 years ago, this group reported that one active rhodopsin can activate close to 1,000 transducins, or GT. Well, over the years, over the decades, this concept has transcended vision to become a textbook dogma about GPCR signaling. Now, in the meantime, within the field of vision, there have been some issues about believing this large number. And just a few years ago, we decided once and for all to solve this problem with live cell recording. And what we found was that the real number is only about 10 to 20. Now, how do we do that? We basically, without going into the details, we were able to, for the first time, to actually obtain the profile of the electrical response produced by a single transducer. We know its amplitude and we know it's time course. So from the ratio between the time-integrated single photon response and the time-integrated single transducing electrical effect, the ratio will give us the number of transductions, and that comes out to be about 10 to 20. By the way, this number actually comes pretty close to what Ashavsky and Marie Burns have published based on some biochemical measurements. Now, the bigger question is, how about Gpc signaling in general? Now, Gpc signaling predominantly is activated by ligands, okay? So in this case, it's highly unusual. It is activated by light. Now, unfortunately, there's not been any equivalent information about this number here for ligand-gated Gpc signaling, except for what we did about 15 years ago. So first, let me talk about, just briefly introduce olfactory transduction. It is very similar to phototransduction in rods and cones, except that cyclic AMP is the second messenger, and it typically goes up with odorant stimulation, unlike in vision, where the cyclic GMP and it always come down in level in response to light. There's some other very important properties of olfactory transduction, such as the involvement of a chloride channel, which is depolarizing when it opens, to serve as another step of amplification. For now, let us just focus on what this number here is, which is the amplification. And in 2005, we found that it is very low. The gain is actually much less than 1. From the Poisson distribution, what this tells us is that most of the time, when this complex is active with the odorant and the receptor, nothing happens. There's no consequence. And only occasionally would the complex successful in activating a GO, which then will go on to produce an electrical response. Now, why is this gain so low? Well, based on our experiments in 2005, we think this is simply because the lifetime of this complex, that is the dwell time of the odorant on the receptor, is very brief, associates very quickly, such that there's little time for this complex to find the downstream GO and activating it. Now, in those earlier days, we encountered the olfactory receptor neurons randomly, and each of them would have a different receptor, odor receptor, of unknown characteristic. At the same time, we use just a few generic odorants for stimulation. So one major concern is that the low gain that we found in these experiments was simply due to the mismatching of the odorant and the receptor. Okay, that is to say, maybe if you use the right odorant for the right receptor, the gain would be very high, just like rods and cones. Well, most recently, we focus on one particular receptor. There's a lot of details here which I won't go into, but at any rate, we can consistently record from olfactory receptor neurons that express only the M71 odorant receptor molecule. And this has been well studied, and the best odorant for this M71 is acetophenol. So we did those experiments, and with biophysics and other sort of both calculations and measurements, we found that surprisingly, even for the best matched complex between M71 and acetophenol, the probability of success remains very low, about 10 to the -4. Okay, so basically, we think that in olfactory transduction, now, of course, we've only done this particular receptor, M71, is actually very difficult because we have to make a special mouse to do these experiments. But the bottom line is, generally speaking, the gain is much less than 1, and we still don't know whether, in fact, this can be generally applied to all odor receptors. Maybe not, especially for molecular pheromones, which have very high specificity for activating transduction. We think, at least for now, that this low gain may be typical or representative of ligand-triggered GPCR signaling. And of course, only time can tell us how general our findings is. In contrast, rod phototransduction has a relatively high amplification here. We think this is actually very unique because of the circumstances that rod signals are used in vision. And we can go to that, come into that in discussion later. But I'll just say that we have some evidence to suggest that for cone phototransduction, which of course is phototransduction, but even but for cones, even this number does not apply. And we think in cone phototransduction, in fact, the gain may well be around 1 or less than 1, although it's impossible to measure that gain for cones. Okay, now let me now describe briefly the single photon response, a concept that I introduced a bit earlier. Now, way back in the 1940s, human psychophysicists already know that in under dark conditions, a dark adapter rod can signal a single photon absorption. Then in 1977, with the suction pipette technique, Beta Lam and I succeeded in resolving these single photon responses as shown by these smallest all or none responses, well resolvable above the background noise level. Now, the important question is, in order for a rod to signal a single photon, of course, a single photon response has to be large enough to be resolved. But in addition, the rhodopsin pigment cannot have a very high spontaneous rate of isomerization in the dark, because then we cannot separate between real light and pseudo light. Now, because each rod expresses so many rhodopsin to the tune of 10 to the 8 to 10 to the 9 molecules, invariably there will be some fake events that would happen sooner or later. And indeed, we were able to see these events in complete darkness, shown by marked by these arrowheads. This is, by the way, from an amphibian rod at 20 degrees centigrade. So after correcting to 37 degrees for, of course, we humans, and after correcting to the human, the rhodopsin content in a human rod, we can find out what the rate of events that would happen in our eyes. And the conclusion is that it'll be on average 1 event per. So the final notice that has been mailed to your resident. So basically, the rate in our own eyes, each rod would have about 1 event in the dark per minute, okay? Now, so you may say, oh, is that enough noise to give problems? Not quite, because it is known that humans can detect a light flash that actually leads to only about 5 or 6 isomerizations, okay? So our eyes are very good. And in fact, if you focus on one particular particular rhodopsin molecule in human eye, the conclusion is that based on calculations, that you have to wait, if you fixate on a particular rhodopsin molecule, you have to wait on average about 100 years at 37 degrees before you see a spontaneous flip-flop So in fact, our rhodopsin is indeed extremely stable. It's just that there's so many of them required for capturing light that we have this, bit of dark noise. And of course, in fact, if you compare the rate that we calculated with what human psychophysicists have measured about the dark noise, they come out to be almost identical. In other words, in dark adaptive conditions, the rhodopsin spontaneous isomerization is what limits the threshold of light detection. Now, what about cone? How about a spontaneous noise in cones? Well, for 30 years after we obtained this result from rods, nothing happened. And that's because you cannot resolve these events in a cone cell. Therefore, we cannot find out how noisy the cone pigment is. Finally, we got a way out. We got around the problem by expressing a cone pigment as a transgene in rods, either xenopus rods or mouse rods, so we can borrow the high amplification of rod phototransduction to detect these events. So in fact, in such manner, we found that the red cone pigment, if expressed human red cone pigment in mouse rods, we obtained a rate of spontaneous activity, which is as high as 100,000 times higher than for rhodopsin here. So this for red pigment. Now, nature also helped us in that there's in amphibians, the blue pigment is expressed in some rods. They're called green rods, so we need not do any transgenics, and we were able to measure the spontaneous activity of blue pigment, and it turns out to be about 200 times lower than rhodopsin. So putting all the results together, here is a plot of the rate constant of spontaneous activity in a log scale against the lambda max of the pigment. So this is A1 blue cone pigment, A1 red cone pigment, and A2 red cone pigment. Of course, A1 and A2 are the chromophores used. A1 used by land-based animals, also marine animals, and also the birds. But A2 is used by some amphibians and aquatic fish. And going from blue pigment to A2 red cone pigment, the noise rate goes up by about 10 million times. Now, let me say that with experiments, we actually obtained a very nice, simple physical relation between the lambda max of a pigment and also the isomerization activation energy. I won't go into it, but it's actually an experimental measurement, which holds true for all pigments that we've looked at. So for any lambda max, we can calculate what the activation energy of isomerization is. And then we plug these numbers into, with statistical mechanics in physical chemistry, we can actually calculate what the rate constant is. Now, we cannot use the Boltzmann distribution, which is still used a lot in biophysics these days, but Boltzmann distribution applies really for an ideal gas. So it doesn't work for polyatomic molecules such as rhodopsin. But here's this person, Hinshelwood, who actually got a Nobel Prize in physical chemistry. He's a British physical chemist, and he was able to extend the Boltzmann distribution into one that applies to polyatomic molecule. I won't write out the mathematics because it's quite complex. But the bottom line is, with this distribution, we can calculate without any free parameter to obtain this smooth curve, which actually fits the data quite well. Now, why are there no points here? Well, simple, because in fact, in nature, there isn't so far any pigment known to have a lambda max longer than 620 nanometers. So that's all we can do is up to this point. And all of this, of course, is theoretical, but we think that this relation, in fact, for the first time in 50 years, we were able to describe the behavior of the spontaneous activity of pigments really for the first time. And but by extrapolation, you can see that if you had a pigment with a lambda max over 620 nanometer, it's going to have much, much more noise, about 360 times more. And that may be the reason why nature does not actually use pigments that have lambda max in this region, because then the pigment is essentially useless with so much spontaneous activity going on. So this is the Boltzmann distribution, which in fact has been used, for the last, 50, 70 years by people to describe pigment behavior, and it doesn't work. And that's why, in fact, the problem was in a standstill until actually, I would like to mention Ayla Lorilla's name, who was not quite a postdoc in my lab, but we worked so well together when he was very young, and so I view him as a postdoc. But he was the first one to go back and use the Hinshaw distribution, even though way back in the 1940s, that somebody who was a postdoc of a very famous physical chemist at Caltech, first used, but then it kind of disappeared. But the bottom line is that we can describe this behavior very well with this distribution. Now, these are the cones. What about rhodopsin? Well, of course, rhodopsin all cluster around this lambda max. And basically, the conclusion is that for a given lambda max, the rhodopsin is about 25 times less active than the corresponding cone pigment. And this makes sense because it's well known from chemists that the binding pocket for the chromophore is closed, and therefore probably much more resistant to vibrations of the molecule. And that's the reason why I think that you have a 25-fold lower activity. And in terms of the mathematics, it's simply to say that this so-called pre-exponential factor in physical chemistry is about 25 times lower, okay? That's how we describe it. Now, most recently, we were able to obtain this spontaneous activity from two more pigments. This is the mouse green pigment, and this is actually a man-made rhodopsin, deliberately shifting the lambda max by about 20 nanometer from the normal rhodopsin. And indeed, even these data fit very well to the corresponding rhodopsin, cone pigment, and rhodopsin. All right. Now, let me spend the remaining time on non-image vision. So basically, around year 2000, A.K. Provencio, at the time at Uniform Health Sciences University, and now at the University of Virginia, he cloned a new pigment from frog skin melanophores. These are skin cells that would turn dark when the animal is in a dark background, and it turns light when it's a light background. It's an opsin, and furthermore, they found that the melanopsin is not just in the skin cells of the frog, but also in the inner retina of the amphibian, not just amphibians, but even in a mammal. So taking that cue, David Burson at Brown University then labeled retinal ganglion cells from the suprachiasmatic nucleus, which is the nucleus that underlies the circadian rhythm in animals. And then they record from these ganglion cells, and they found that indeed, these cells respond to light even if you completely block all synaptic transmission in the retina. Now, for our part, we cloned rat melanopsin and we made an antibody. And this is a flat mount rat retina. And the green color is the immunofluorescence for rat melanopsin. Basically, the pigment is present in the cell body in all the dendrites down to the fine ramifications and including the axon. Now, we can follow the axon all the way to the optic disc. Therefore, these are ganglion cells and not amacrine cells. Basically, you can count up all the cells, and we found that it amounts to a couple of thousand or several thousand of these ganglion cells, which is equal to about 1 to a few % of the total retinal ganglion cell population in humans and in mouse, rat, and all that. Now, we then collaborated with David Burson and showed that these cells that express melanopsin indeed are intrinsically light sensitive. Namely, you can block out all synaptic transmission and these cells still respond to light. Whereas if you knock out melanopsin, which we did from mouse, these cells lose the light sensitivity, okay? Now, where do these cells go to exactly in the brain? Now, unfortunately, we cannot follow this immunolabeling because this disappears once the axon leaves the eyeball. So melanopsin is not present in the axon except for the part when it's crossing over the retina. So what we did to trace the target is to make another mouse, which is a tau-lec-Z knock-in, knockout animal, namely replacing melanopsin with the tau-lec-Z marker gene, which you can then use X-gal as a substrate to reveal the cause of this axon, as shown here. So this is a tau-lec-Z knock-in, knockout animal. Is a heterozygous, I think, this one. And we can trace every single axon to the brain, and it's given by this figure here. This is a sagittal section of the mouse brain, and all these nuclei, more than a dozen of them, receive innervation from these melanopsin ganglion cells. Most of them are for non-image vision, such as the SCN, the suprachiasmatic nucleus, the IGL, which is a nucleus that controls the pupillary light reflex, and many others. Although at the same time, I'm sorry, not IGL, OPN. OPN is a nucleus that controls the pupillary light reflex. But at the same time, there's also innervation of the image vision nuclei, like the dorsolateral geniculus and also superior collicus, which is involved in saccades of eyes. And this is the suprachiasmatic nucleus. You can see the very, very dense innervation by the axons. And this is the OPN, olfactory protective nucleus, involved in the pupil constriction. Now it's known that there are at least 6 types of these melanopsin ganglion cells, M1 to M6. And with biophysical experiments done by Michael Doe, who was a postdoc in the lab, we found that actually even the density of melanopsin is very low in M1 cells, to the tune of only 10,000th that of the rod and cone pigment, okay? But interestingly, even though the low pigment content and therefore the low photon capture, these cells have a very high ability to transduce the absorbed light, and in fact, they can also signal single photon absorption, just like rods. Oh, just here is to show you, we've also cloned human melanopsin. I think I described it kind of in passing earlier. and we make another antibody against the human melanopsin, and this is a human melanopsin cell, and this is a primate, a macaque monkey melanopsin cell. Now, this observation explains a paradox that existed in clinical ophthalmology, namely that human beings who are unable to see because they have lost the rods and cones from disease, but yet they can still somehow sense the passage of the day. They know when it's time to go to bed, or when they fly across time zones, they know that they need to, or rather they can get used to the time lag. And that's because that these cells are still around, even though rods and cones are no longer there, to mediate the non-image vision. So now, I just want to quickly show you this, namely that the black traces here is an M1 mouse melanopsin cell, and these black traces are the responses to individual dim light trials. And the red traces are what we calculated to be the single photon response from these cells. And you see that the black traces from individual trials, either no response or response, no response or response. So this is exactly like what we observe with rod photoreceptors. Interestingly, the amplitude of these responses here are twice as large as that for the mouse rod. So they're quite big. And yet, the kinetics is very slow. They're about 20 times slower in kinetics than that of rods. And of course, these cells are mostly for non-image vision, which does not need the very high temporal resolution. And in fact, it is very good for summation of signals over time. So this is one unusual characteristic of these responses. Now, what is the transduction mechanism? This, of course, is what we're always interested in, given my background. And just to remind you, these are the rods and cones, and it uses a cyclic GMP pathway, shown here. What about M1 cells? We solved this problem a few years ago. This was 2018. I couldn't see here, but it's about 3 years ago. And basically, the mechanism is exactly identical to that found in the fly photoreceptors, which are rhabdomeric photoreceptors. So unlike rods and cones, which are ciliary photoreceptors, the rhabdomeric photoreceptor uses microvilli. And of course, just like in fly, PLC beta 4 is a key enzyme for this phototransduction, and trip channels to be the final step that gives rise to an electrical response. And by extension, one would expect that the upstream G protein would be the GQ subfamily. Now, of course, the 4 members in mammals for the GQ subfamily, namely Q, 11, 14, and 15. And it took us several years to figure out which of these is the right one. And it turns out that there are 3 of them that all involve in this mechanism. You can delete 2 of them and still phototransduction goes on. So you can imagine how much work it takes to get to this result. In fact, that the genes for 2 of these are also linked in the same chromosome. So it is a nightmare to solve it, but we did. Okay. Now, interestingly, even though this is equal to or the same as the rhabdomeric phototransduction mechanism in the fly, in melanopsin ganglion cells, even if you use an electron microscope, you're not going to see any microvilli or, for that matter, cilia in the cell. So this is very interesting. All right. Now, what about the other subtypes? I'll just tell you something I can't see here because of all these bars, but I think you probably be able to see it. So this is the M4. Let me just describe M4 first. Basically, the channels that we found to be involved in phototransduction, they call Hcn channels, hyperpolarization activated or cyclic nucleotide activated. That's why they call Hcn channels. So the gating is fairly complex. In fact, these channels are very important for the heartbeat. They also call IH current, because if you delete this, the heart will stop beating. I mean, this is the channel that gives rise to the pacemaking activity in the heart. Now, when this paper was published in 2018, we haven't really solved everything. We know the cyclic nucleotide that would activate the channels based on our experiments, but we do not know what is cyclic A or cyclic G, okay? And for that matter, we also don't know exactly what are the upstream steps. Now, Hcn channels, by the way, in the heart is mediated by cyclic AMP, the modulation. But we cannot use that argument for our work here because we know, for example, that in olfaction, even though the CNT channels mediate olfaction is more sensitive to cyclic G than the cyclic A, in fact, is the native second messenger. So we cannot use the argument regarding the sensitivity of these channels to derive what the second messenger is. Now, what about M2 cells? Well, very interesting that each cell we found, within each cell, both pathways are at work, both triggered by melanopsin. One pathway is the one like M1, and the other one is like M2, M4. So actually, this is the first example found in nature of a photoreceptor that uses both the microvillus transduction mechanism as well as the ciliary phototransduction mechanism, kind of like rods and cones, except, of course, that the channels are not CNT channels. Now, 3 years later, we know a lot more about these mechanisms, and basically, it is indeed cyclic AMP, all right? And the upstream enzyme, we think, is an adenocyclase. Unfortunately, there are about 9 to 10 adenocyclases, so it's very difficult to figure this out, but we are in the middle of figuring it out, actually. Now, a couple of really interesting inferences can be made here. First, that it is known for ciliary phototransduction, cyclic GMP is always the second messenger involved in transduction. Whether you talk about C. elegans or us or any other animal that has been looked at, the photoreceptors, if the ciliary photoreceptors is always cyclic GMP, except for one unusual example, which is in the jellyfish. In jellyfish, the ciliary photoreceptor actually uses cyclic AMP. So this is like jellyfish. Now, jellyfish is a very ancient animal. If you look at evolution, the jellyfish kind of splits off on its own, whereas all the other animals follow the same evolutionary stream. So that suggests that maybe these cells are very ancient, the melanopsin ganglion cells. But there's another piece of evidence. Basically, I've told you before that in all extent, meaning existing currently in the world, all photoreceptors either use this pathway or this pathway, but not both together in the same cell. But among evil biologists like Walter Gehring, who passed away, unfortunately, he's Swiss, he somehow speculated that maybe the very earliest photoreceptor in evolution actually uses both pathways, but very quickly, these two pathways segregate to different cell types, whether it's microvillus or ciliary. But yet we found these cells in us, the most advanced of all animals. But basically, based on the cyclic A evidence and the coexistence of two pathways in the same cell, we think these cells are evolutionary among the most ancient, but they're still present in us. All right, so hopefully soon I can tell you all the details of these steps. Still, at the time when we published the paper in 2018, it's not clear. Now, so the question is that it? We have in our eyes Rhodopsin cones and also ganglion cells that are intrinsically photosensitive. Well, in our eyes, that's probably true in the retina. But in fish, we found back about 11 years ago or 12 years ago that horizontal cells, these are interneurons in the retina. And we found that specifically the cone-driven horizontal cells, in fact, has intrinsic photosensitivity. And these are dissociated cells, so we define sensitivity, they cannot come from other cells. So here are recordings from these dissociated cells, and what we found was the cone horizontal cells are indeed photosensitive. I won't go into the mechanism, but it involves L-type calcium channels, and the driving pigment is also melanopsin. Okay, so is that it then? We have Rhodopsin cones, ganglion cells, and now horizontal cells, although not in us, that are light sensitive. This is, by the way, catfish, but in goldfish as well, we found the same thing, that some of the interneurons are intrinsically photosensitive. Well, not quite. We found another clear photoreceptor in the eye. And this is actually a discovery by accident. In fact, it is a rediscovery of a phenomenon that has been published something like, I don't know, 30 years ago, although the journal is rather obscure, and we're not aware of this discovery until we actually come to write up our work, and then we search, and we found this. Basically, the point is that the iris in many animals have, in fact, a pigment in it, so that you can just take out the eye, or for that matter, you can just take out the iris finger muscle from the eye. You can hook them onto a strain gauge, and you shine light, and you see a contraction of the muscle. If you look at the lambda max, it is exactly where melanopsin would be. If you knock out melanopsin, you no longer see this intrinsic photosensitivity of the iris muscle. And if you knock out PLC beta 4, one of the enzymes involved in the M1 subtype of ganglion cell, you also remove the response. Finally, using genetics, we can actually observe the presence of melanopsin in subset of the iris finger muscle, the smooth muscle, by the way, shown by the red here. So not every muscle fiber has melanopsin, but because the extensive gap junctions across the muscle fibers, when these are active, they would give rise to a uniform constriction of the pupil. Now, we've looked at the distribution of this phenomenon in animals, and we found that it's literally present in all crepuscular subprimates. Crepuscular meaning they're active in dawn and dusk, okay, but not in daylight. And such as, well, literally, we look at about 10 species, and they're all sensitive, intrinsically sensitive to light, just the muscle by itself. However, for subprimate mammals, which are diurnal, we don't find a phenomenon, okay? And for humans or other primates, whether nocturnal or diurnal, they don't have this phenomenon. Now, this may be a good thing that we don't have it, because doctors, as you know, when they check up on a patient in comatose, a simple way to check whether the brain is still alive is to shine a beam of light into the eye and to see whether you get a reflex. And of course, that depends, you know, before we found this, to be really relying on the circuits that go through the auricular protective nucleus. So It just so happens that conveniently, we don't have this phenomenon so that the doctor can still use this test. Now, evolution-wise, this is actually evolution phenomenon in that suppose if you look at amphibians and you look at the pupil reflex, it turns out people look at that and they found the reflex to be totally coming from the iris muscle, not from anywhere in the brain. So it looks like in lower animals, this is in fact what is the main reason for the reflex. But then over evolution, it gradually disappears, such that, you know, by the time we get to the mouse has both the reflex through the brain, which is a predominant one, and a minor one given by this phenomenon. By the time it gets to us in evolution, the phenomenon is gone, even though we can still observe by PCR melanopsin in the muscle, in the muscle. Well, thank you so much. I think I've overused my time, but these are all the collaborators that I've had over the years, many by providing mice to us. And make sure I don't forget, Dennis Baylor and Alan Hopkins are my two mentors, and I learn a lot from them. And these are the people who I've worked with over the years. These are all the postdoctoral fellows, and these are the graduate students, and the ones in STAR is current members. Thank you so much.
Okay, everybody, if you have any question, please send me just note through chat that you would like to ask the question. And if you're in our conference rooms, just maybe shout out of your question directly. So King, before people get their heads together, let me ask you a question related to your work on goldfish, where you show those three pigments, red, green, and blue, that they have a different, I would say, the dissociation rate of the chromophore, right? That the blue do not dissociate.
It's not so much the rate, but it's the equilibrium. That is to say, the red pigment dissociates even in equilibrium. Now, let me just say something, a detail. We are very careful when we do these experiments in goldfish. We can afford to because we would kill the fish at time 0 and very quickly take out the retina, and then we time the time lapse, you know, from that point on, and then we quickly do recordings, and we also do microspectrophotometry. This is done by Carter Cornwall at Boston University. So we want to make sure that in fact the phenomenon is not an artifact. And what we found was that and then the way we find out how much is naked pigment is by adding chromophore afterwards to see how much increase in the optical density there is in the cones. And we also use recordings to check the photosensitivity, and they both checked out pretty well. So it looks like that this equilibrium already exists in situ in the cells. Of course, in situ, you have a supply of chromophore from the RPE and from the mural glial cells. So we think that this phenomenon is actually a true phenomenon that happens in the live animal. Now, we are doing other species right now, and hopefully we're going to check out. We're actually checking out in primate, which of course is a major undertaking because they're expensive and they're not easy to get. But we are doing them right now.
So if you use microspectrophotometry, aren't you not bleaching with the light from spectrophotometer any of the pigment, or how could you exclude that you just bleaching it during the measurement?
Well, basically, now this is not my domain. This is Carter Cornwall, whom I respect enormously. Carter was also Vladimir's PhD advisor. If somebody else is doing it, I might have doubt, but Carter, you know, I emphasize it is extremely important to make sure you don't bleach the pigment while you're doing microspectrophotometry. And they've done the whatever controls. The paper's already published in Current Biology last year. So, but yes, I mean, we think this is real now. I should emphasize that Vladimir Kefelov, while he was in my lab, also did some of these experiments, but we used a different procedure. We didn't use quite the details that I just described. We simply use the sensitivity as a measure of the presence of opsin, and it's not as direct as using spectrophotometry and also in a goldfish. It turns out that the sensitivity is not that sensitive to the presence of opsin. So if it's only like 1%, 2%, you cannot, or even 10%, you cannot actually get very reliable measurements. But I suggest that whoever is interested in go to read our paper.
Thank you. Vladimir, you had a question.
Yes, thank you, Christian. Yeah, I have an echo here. Thank you, King. Beautiful presentation and just a real tour de force. I was wondering whether in the spirit of this general presentation...
Perhaps just mute your laptop.
Vladimir, mute your laptop. But then the room is unmute.
Yeah, mute the laptop, but unmute the...
Okay, can you hear me now? Yes. Okay, so thank you again. Thank you, King. Beautiful, real tour de force of just an amazing body of work over so many decades. In the spirit of this general presentation, I was wondering whether you would care to tell us your thoughts about some of the big remaining questions in the field that you think we should be working on over the next 10, 20 years?
Well, you know what? I'll be dead in 20 years, so it's not really. Even if I tell you I couldn't find out what is what is true, what is not true in the next 20 years. You know what? That's a very interesting question. I can tell you what I'm working on. I mean, if that helps at all. We are, of course, pursuing several questions. As I mentioned before, I don't like joining the crowds. I like to do something that very few people work on, so I can take my time to do it. So right now, we are working on UV pigment. And we already make multiple mouse lines to look at UV pigment. Now, what are we interested in? Well, we're really interested, I mentioned before about the spontaneous activity, which you have done a lot on while in my lab. Our question, and of course, the theory that we propose, which is really the first time in more than 50 years that we understand exactly how these pigments behave. And we want to know whether we extend the theory to UV pigments. My guess is it would. But it's very interesting because UV pigment, as you may know, that the shift base, is I'm talking about the bond between the lysine and the shift base is unprotonated. So we want to know whether what the protonation would do to the spontaneous activity. We already got some data, but it's still early. Okay, so that's one question. I don't think anybody's working on it. Another question we're working on is diseases. So we saw the D190N. And now that you ask me, I want to be very honest. You told me, doesn't matter. I mean, there's nothing wrong with that. You wrote me an email saying that you work on the G90D. And you like our paper, and you said it would be a very nice approach. By the time you told me, we already decided to make a mouse. But there will be differences. So I don't know whether you've got the answer. If you already got the answer, good for you. Because we are we are still, in the middle of it. But we're going to do it rather differently from what other people are doing. So even if we've got both labs are doing it, it's okay because we're not using the same strategy. So yeah, the G90D, of course, that is an answer that everybody wants to get. It's been worked on so much in the last 20 years, but we have our own theory, but let's just see. And we also work on some other diseases. Will I live long enough to see the result? I don't know. What else? Well, now this is really half a joke, but it's not a joke. We're also interested in myopia. Why are we interested in it? Because we discovered, well, let's put it this way. I guess some people in the room have heard of the Star-Crawford effect, which is the orientation of photoreceptors depending on the location of those photoreceptors in the retina. That is to say, those photoreceptors at the very back of the eye are perpendicular orientation to the visual axis, right? Or if they point towards the center of the eyeball. But for photoreceptors that are located at the interior region of the retina, they are tilted towards the pupil of the eye. So they're no longer perpendicular to the surface of the retina, as those at the very back of the eye would be. And this problem is 90 years old, 90. And there's no data on it except by Alan Lattice, who did some anatomy and showed that in fact, yes, because the Star-Crawford effect originally was a human psychophysical phenomenon. But Lattice first did some morphology, and he found, in fact, the tilt as a function of the distance from the fovea, for example. We actually work on mouse, and we published a paper last year in Science Advances. And in fact, we obtained what we think is a correlate of the human Star-Crawford effect. That is, if you bred mice completely in darkness, never saw a photon of light, their photoreceptors are tilted, strangely. And we just want to figure out what's going on. And because we began to work on the periphery of the eye, we began to think about myopia. And this is more a dream than anything. I mean, because myopia is like 300 years old. And still, people don't know what on earth is going on that makes the eye elongate. So I may be being very naive, but at least I can work on something that nobody works on, which is what I like. Anything else? Well, there may be something else, but maybe I just forgot. So no, I'm serious. That's all I work on. What other questions?
All right, so let's go to Steve.
Okay.
King, how are you?
Steve who?
Steve Fleisler in Buffalo, New York.
Oh, Steve, hello, how are you?
Good to see you.
Same here.
I always enjoy your talks. I learned a lot today. And one of the things that I was ignorant about, the many things, is the difference between the olfactory system and the, let's say, the rod visual system. And you showed us that the odorant receptor, when it's activated, apparently is very slow to couple to its cognate G protein. So the amplification is 1 or less. Did I get that correct?
I wouldn't call it slow. I would rather say that the reason why the amplification is so low is that the lifetime of the odorant with the receptor complex lasts a very, very short time. In fact, we got some measurements back in 2005. This is the time when we still are not focusing on one particular receptor. And we can measure the lifetime. And that's of the order at most a millisecond. That is, the odorant binds to the receptor, and within a millisecond, it dissociates. And just no time for the complex to go and find and activate the G protein G alpha O. So now, in fact, you can connect that to vision because, in fact, why does the group, headed up by Stryer way back 40 years ago, how did they calculate the number 1,000? It's actually pretty, well, I would say pretty simplistic. They simply take the rate of activation of transduction by rhodopsin and multiply that by the lifetime of rhodopsin, active rhodopsin. So in those days, which is quite early, they simply use the time course of the single photon response, which lasts about a second, at least for amphibians, and they get some kind of a rate of the activation of transduction by rhodopsin is of the order maybe 100 per second, and so, or maybe 1,000. I don't know. I mean, the way I think it's probably 1,000. Of course, those numbers actually are very, in those early days, very, very iffy. But I think the 1,000 really comes from 1 second multiplied by, which is the lifetime of rhodopsin, which they believe, which of course is wrong, multiplied by the rate of activation of transduction, which is about 1,000 per second. And that's how they get 1,000. Now, of course, the lifetime of rhodopsin in mouse has been measured, I would say mostly by Marie Burns' group at UC Davis. And the time is of the order of, I think the latest is of the order of 40 milliseconds. And if you multiply 40 milliseconds of the rhodopsin lifetime by the rate of activation of transduction, which I think they got numbers of the order of about 120 per second. So you will get about like, I don't know, 40, 50 transduction, which is actually not far off from what we calculated based on the transduction. We can actually measure the single transduction effect. So from the way I described it, you can get a number which is very close to 10 to 20. So in short, it is the lifetime of the GPCR that would in some way dictate the number of G proteins that the GPCR can activate, at least as a first approximation. Did that make sense?
Yes, it does. My question was because you did early on explain that there's this very rapid dissociation of the odorant from the receptor. I was wondering if there was a second order effect that was biophysical, the lower coupling to its cognate G protein.
Olfaction, it's interesting. In the same olfaction, there's not a lot of course, molecular understanding is known from the work of Richard Exel and Linda Bug and their associated people over the years. But in physiology, there's really not that much which is known. But I would say based on what we understand with our physiology, I don't think you need to hypothesize yet another mechanism. In fact, we did try to really learn more about the dwell time of the odorant on the receptor, but it's just too short. Our experiments have a certain limitation. We cannot actually resolve. Actually, 1 millisecond is really at the limit of our resolution. But I don't think you need to have another step or some other factor that would underlie the G protein, the number G protein. Actually, we are continuing with this work. And in fact, again, rather than asking me what we think the interesting questions are, I think one question I really want to know the answer of is exactly what is this gain for other GPCR signaling? Because nobody has ever published any data like what we have done. And I can tell you that we are not just looking at olfaction, which is a great prep, but also looking at, we're looking at the heart. Because what is the one of the best known GPCR signaling pathways? Of course, one is the rod, The other is the beta adrenergic receptor in the heart. We are working on that right now. And of course, I did mention in passing during the lecture that we think cone photoreceptors are quite different from rods. First, let me say, why do rods behave the way it does, which I think is very unusual and kind of a unique. And that is, rods are designed for signaling single photons. So they have to develop a mechanism so that they can signal a single photon absorption, which means that a single active rhodopsin has to produce a signal large enough for them to signal that electrical signal to this downstream cell. And so they have a pretty high gain, which is about 10 to 20. And they have to do that because each rod has a signal. Now, in a typical case, like a GPCR signaling, I don't think you need to have single GPCR molecule signal. It's not necessary. It's just like ion channels. You don't need, you don't need every single ion channel molecule of a particular type to open. It's only kind of an ensemble effect. So as long as some of the GPCRs of a particular type are going to work, that's good enough. So in fact, for cones, we think, now we don't have the absolute evidence for it, but we think, based on inference, we work on goldfish. We know that goldfish red cones has a incredibly low sensitivity. It's about 500 times lower than that for green cones. So if green cone is only like 10 transducents, the red cones must be much lower than that, much less than 1. But at this point, we cannot directly observe them, but that's my thinking. So rods are very unusual, okay? Even for cones, it's very different from rods. And so, so the main question is, you don't need a high gain as such, because typically, like in the heart, when you activate the beta-1 adrenergic receptor with noradrenaline, I mean, not every single beta-1 adrenergic receptor have to be active, as long as enough is active. And of course, in the nose, you say, well, so the gain is so low, how can that work? Well, no problem, because I'm talking about per receptor molecule. My question is, if you look at one particular odor receptor molecule, the probability is extremely low. But there are a lot of receptors on the cell. In fact, we actually have, the paper is going to hopefully come out before too long. We actually, for the first time, measure how many receptor molecules for the odorant are there in a odorant receptor cell. It's of the order of 10 to the 5. Okay, this is based on very precise biochemistry. We got help from Baudet on this, and so there are a lot of receptors there. Okay, and that would multiply up into a high gain. And furthermore, we know that in the olfactory system, in the nose, for any particular odor receptor, there are of course over 1,000 of them in the mouse and about 250 in the human. There are a lot of cells all expressing the same odor receptor. And then they project their axons, every single one of them, to the same synaptic spot in the olfactory bulb called the glomeruli. So if you multiply out the 10 to minus 4 by a factor of 10 to the 5th, actually much higher than that, that's per cell. And you multiply by the number of cells, it's going to be a large number. So there's absolutely no problem of the signal being too small. There'll be plenty of signal to go to the brain. Am I making some sense?
Yes, but I still have a problem with this concept because the odorant will come with the rate of diffusion. And if there is a high affinity for any of the odor, then you will have to have a slow dissociation. Otherwise, you will not have a high affinity.
Oh,
So,
well,
so
hold on.
I don't know how you can reconcile that. It's puzzling for me.
Well, I would say that, yes, high affinity. Well, how high is the high? Actually, okay, concentrating on the nose. You don't want to have very high affinity for the odor receptors because to follow a trail of an odorant, you cannot have, high affinity because then whatever is bound never comes off. And how can you actually follow a gradient, right? So we don't think now. Exceptions such as pheromones, I cannot speak for that. Actually, we are working on a pheromone receptor now, and we will have some idea what the situation is like for a pheromone. But I don't think there's any. I don't think you can have. I mean, the bottom line is we are. Obviously, until you see it, I would say this. This acetophenone, we still do not know what is the dwell time of acetophenone, which is, of course, one of the most potent odorants for the M71 receptor. We wanted to measure it, but we could not, just because our apparatus, this is not light. I mean, you have to, you can give light very briefly. To apply odorant, it involves a lot of mechanical things that we need in the setup. So we cannot resolve the dwell time of acetophenone on the M71 odorant receptor. That's all I can say. And the probability of success, 10 to minus 4, we actually use physics and diffusion. But,
All
only
right.
to, yeah.
We will be looking for the publication. There is a question from somebody called C.
Okay.
I don't know the name because it says C.
Yes, hi. Hi, Professor. My name is Chris Sander. I'm in the Bolshevsky and Kaiser labs.
Okay.
I just had a question on the olfactory receptors that you've mentioned. If animals that are more reliant upon their sense of smell produce orthologs with higher signal gain, or if they just have more receptors.
Okay. You mean when an animal that relies on smell, very importantly, do they have more odor receptors? Is that what you meant?
Yeah, well, I mean like like say a dog or something, like a bomb sniffing dog. They can smell these things that we can't. And I was wondering if they have different orthologs.
Actually, you know what? Of course, I've always been wondering about this question. Even though I work on olfaction only really a quarter of the time. Well, I happen to have read, I love dogs, but because of my allergy, I can only read it in a book, but not really touch them. It turns out, in one of these dog books that I've read, they claim that a bloodhound, the olfactory epithelium in a bloodhound's nose, when you stretch it out, is larger than the skin surface of an entire animal. Okay? Now, I just want to say again that you don't really want to have very high affinity because how many odors do we smell? I mean, countless. In fact, there's no limit to it. I mean, there are a lot of things that you may never come across, but you can smell them pretty well, right? So I just don't think that, so basically you have about of the order of 1,000 odor receptors in the mouth. And that may be a better example than human because we're not exactly use our nose for everything. So you need, there must be tens of thousands of odorants. So each receptor, in fact, is known in the field that a lot of receptors are very broad sensors. So in the mouth, we know in the old days when we just focus on, we just don't care about the receptor itself, we just encounter randomly any receptor cell, you can just put a few odorants. I would say 1 in 10 of these odorants would activate them. So a lot of them are very broad sensors. There are occasional, very narrow sensors. But the bottom line is that most of these guys have to pick up a lot of stuff. And of course, I mean, how many smells how many individual smells are there in a dish? Many. And you know, it's the whole conglomerate of the response that give you the function, right? I mean, you're not.
Yeah, so it's more of a cell number kind of thing then. Yeah.
Well, it's actually a combinatoric event that is
Okay.
that each, you know, basically each cell can smell many things, and then there are many different kinds of cells. It's kind of like vision, except that instead of having just rhodopsin to sense, you know, for dim light vision and 3-cone pigment to sense the other colors, you're talking about thousands of these, and each one is very broad. I mean, of course, in vision, it is the energy of the photon that really dictates, you know, how well the stimulus would activate a rod or cone cell. But here, you need to have the molecule, you know, precise structure and geometry. Actually, it's not, you know what, it's absolutely not known how the odor receptors are going to sense a particular odor. And I would say that, Vladimir, you know, one way to do it is to switch to olfaction. And that would be a very challenging problem to go into, but it's very interesting.
We have also in our audience Bob Mulday. Bob, are you there?
Bob, yes, I saw Bob somehow, somewhere.
Bob, unmute yourself.
Yeah, I'm here.
All right, Bob, thank you for joining us. So the final comments, we had a great conversation. And the final comments goes to Bob Mulday.
Hi, King. How you
Hi,
doing?
Bob.
Hi, Chris.
So
Well, actually, I should say that the latest olfaction work was done in collaboration with Bob Mulday. It's not out yet, but yeah.
all right, King, thank you very much. Bob looks like he cannot get his mind together to make a final comments, but we'll be working on him. So
Well, I
thank
can
you
make
very much.
I can make a minor comment, but the olfactory of the insect has been recently examined, the structure and everything, and certain insects only have maybe a handful of olfactory receptors, which are actually coupled to ion transport directly and don't use the G protein coupled receptor mechanism. But analysis of the structure looks like it's evolved so that it can take on a lot of different odorants in the pocket. And so I think it's designed specifically for low affinity interaction between the odor and the receptor. And I think that may be different than the epinephrine and adrenal receptor. So I think we'll have to see as time goes on as to whether there is any common transduction mechanism and amplification.
All right, with that, thank you very much, everybody. Thank you, Bob, for your comments. Until next week.
Thank you very much. Take care. Bye.